Okay, so now let's look at carbohydrates. So the chemical composition in carbohydrates is carbon, hydrogen, and oxygen. So if asked that, you could also say CHO. The structure, the main structure of the carbohydrates is the formula of CX, open your brackets, H2O, close your brackets, Y, where Y is going to equal to X, and you need to know that general composition. So monosaccharides joined together will form a carbohydrate. Now the basic unit of carbohydrates is glucose. Glucose is an example of a monosaccharide. Mono being one, saccharide a sugar unit, one sugar unit. And the chemical formula for glucose, which you need to know, is C6H12O6. So there's a fixed ratio of 2 hydrogens to 1 oxygen in the carbohydrate. So there are different types of carbohydrates that we need to know. Monosaccharides, so mono being one, it's a single sugar unit. And we need to know an example of a monosaccharide. So here we have glucose, fructose, or galactose. So glucose is what I like teaching. And then we need to know a source of this monosaccharide. Fruit, sweets, chocolate. Also that it's soluble in water. Now with your monosaccharide, there is an experiment, and the experiment is to test for a reducing sugar. So with this, the reagent that we're going to use is Benedict's, and what we're going to look at is glucose. So to glucose, we're going to add Benedict's solution. Our control will be water, and to the water, we would add Benedict's solution. So at first, it's going to be blue. So to both, you're going to place in a water bath at 60 degrees for 10 minutes. A positive test for a reducing sugar is it goes from blue to brick red. Our control will stay blue. Disaccharides. Disaccharides consist of 2 sugar units joined together. You also need to know an example of a disaccharide. This example I'm teaching you is maltose. Maltose is it's a glucose and a glucose joined together. So maltose is made from glucose and glucose. Okay, one glucose to another glucose. It is also soluble in water. Now a polysaccharide. Poly means many. So a polysaccharide consists of many sugar units in a chain. And an example of polysaccharide, we have starch, we have glycogen, and we have cellulose. Chitin, so that builds up the cell walls of fungi. It also helps to form skeleton of insects and crabs. Now a source of this starch would be from bread, pasta, potato. So that's your source of a polysaccharide. It is insoluble in water. And with that, there's an experiment to test for the presence of starch. So the reagent, the chemical that we're going to use is iodine. And you're going to add that to a food sample. Example, a food sample. So it could be a potato, it could be a biscuit, it could be a piece of bread. So a positive test, it's going to go blue-black is your answer for a positive test for iodine. Your control, so if you got your potato and put a few drops of water, the control will stay the same colour of the iodine, so a yellow-brown colour. So starch. So starch is many glucose units joined together. It can be easily broken down. Glycogen. Glycogen is a storage carbohydrate. So it's a storage carbohydrate in animals, and it's mainly found in liver and muscle. Cellulose, also known as fibre, cannot be digested by humans. It adds bulk to the diet, but it can also form cell wall in plant cells. Now we need to know an example of a structural role and a metabolic role of carbohydrates. A structural role of carbohydrates would be cellulose, forming cell walls in plants. A metabolic role of carbohydrates, we could have glucose provides energy when it's broken down during respiration. Glycogen can store energy in animals, and starch can store energy in plants.